So in this part of the presentation, we will be talking about the hepatitis A virus replication cycle. First things first, how does hepatitis A enter the cell? Well, it uses an endocytic pathway, which means that it is able to fuse into the plasma membrane and then bud off into vesicles that are transported into the cell's interior. We talked about it previously, but there are two kinds of hepatitis A viral particles. One of them is the naked without a shell or protective coating, and then the other one is the quasi-enveloped hepatitis A viral particle, which is enveloped. So these do have somewhat different ways of getting into the cell. They do use the endocytic pathway, both of them, but the quasi-enveloped virus is able to travel farther into the cell and delivers the contents to a specialized compartment, the lysosome of the cell, where the viral membrane is degraded and then the RNA is able to release into the cell's interior. This happens slower than with a naked viral particle. So this is an image depicting the course of a quasi-enveloped hepatitis A viral particle into the cell. Okay? Goes into the plasma membrane and then uses a vesicle to travel farther, gets to the lysosome, and then it's degraded, and then it's able to travel. Ultimately, being enveloped helps because it protects the protein from any human response. It's able to be undetected for a longer amount of time in the body, hence why it's able to reach the liver. It's able to reach those hepatocytic cells. This is the genome organization of HAV. You can see it does use IRES like other viruses we've studied in class, for example, polio. The blue shaded part of the genome are the structural proteins, whereas the red and pink shaded region is the non-structural proteins. You can see that the 3C-Pro is a protease that helps hydrolyze peptide bonds, and then the 3D-Pol is the RNA-dependent RNA polymerase, which helps catalyze the replication of the RNA. This is just going to summarize what I just told you in the previous slide. It is 5' cap independent, so that means it does not need a 5' cap to be recognized for translation. Okay, so usually when someone is infected with hepatitis A, it's just supportive treatments. It most of the time will resolve on its own and with no hospitalization necessary. And the way that doctors or scientists are able to diagnose hepatitis A is using serological testing to detect IgM, which is the antibody that is present with recent infection. There are a couple different kinds of vaccine, and the vaccine is meant to prevent HAV before exposure. So there are mostly inactivated, attenuated HAV strains. The examples that I have are Havrix and Vacta. These are 2 shots given 6 months apart, and they help prevent the infection. There is a combination vaccine for both Hep A and B that combines the two, and it's given 3 shots given 6 months apart. There is one other kind of vaccine that I didn't include on the slide, but it is a live attenuated HAV strain, and it's given usually in specific areas of China or India. This is 95% efficient in preventing HAV for 3 years. Okay, here you can see that this is the typical serological course of a hepatitis A viral infection. You can see that HAV particles are actually found in the feces even before any symptoms start. And once symptoms do start, you can detect the amount of ALT, which is a liver enzyme, in the blood to test for liver damage in a patient. But you can see once your symptoms start, you do start to get those IgM antibodies, which do test for recent infection. They tend to fall about 5.5 to 6 months post-infection. However, there is the IgG antibody, which is present with any past infection, or if you get an immunization, that's what they check for in the titers. After infection, you do have a lifelong immunity to hepatitis A virus, as you can see in this blue line right here. It is continuing